Welcome to the Deep Dive, the shortcut to being well-informed. Today, we are strapping in for a journey back to 1944, a time of immense global upheaval.
And we're going to unpack a book that really became a watershed moment in science, Erwin Schrödinger's What is Life?
This is such a remarkable text.
It really is. It's one of the most cited, but I think you could argue it's also one of the most philosophically misunderstood scientific works of the last century.
And it's so concise, so accessible.
Which was key. And, crucially, the author was Schrödinger, a Nobel laureate physicist famous for his work on wave mechanics. The sheer prestige of the man guaranteed its initial impact.
And that impact was, it was just profound. Our goal here isn't just to go over the famous sound bites that everyone kind of knows.
The periodic crystal, negative entropy.
Exactly. And the call for new laws of physics. We want to really excavate the philosophical engine driving Schrödinger's whole argument.
Why did this foundational physicist step so decisively into biology? And what were the long-term consequences of his fiercely deterministic perspective on the molecular revolution that followed?
You really have to place it in its historical moment to get it, don't you?
You absolutely do. The original lectures were delivered at Trinity College in Dublin in 1943, so right in the middle of World War II.
And he'd left Berlin years earlier because of the Nazis.
That's right. And the lectures themselves, they were a cultural phenomenon. People were so desperate for something intellectual, something stimulating during the war, they jammed the lecture hall, waited in line for hours.
It was science presented by a superstar.
And it created this instant excitement, across all the scientific disciplines.
That immediately explains its sort of mythical status. It wasn't just read by biologists.
Not at all. It was read by everyone. And we know from their own accounts that it fundamentally reoriented the careers of the founding figures of molecular biology.
The accounts are so direct, so striking.
They really are. James Watson, for instance, he spoke very openly about reading What is Life? And instantly becoming, quote, polarized toward finding out the secret of the gene.
And Crick wrote to Schrödinger directly, didn't he?
He did. Right after publishing the double helix paper, he wrote to acknowledge the foundational role the book played. It was like a powerful conceptual magnet. It was drawing in all these brilliant young physicists who were feeling this, a general professional malaise after the war.
It gave them a target.
It articulated the ultimate goal: identifying the gene as a profound physics-solvable problem.
Schrödinger framed the question so clearly that it offered immediate direction and legitimacy to this whole new emerging field. But before we get to the critique, we have to build the foundation. We need to reconstruct his core argument.
Which starts with this deep, unsettling paradox about biological order.
Right, a paradox that stems from what he called the order from disorder principle.
Which is essentially the classical statistical physics view of the world.
Okay, so let's unpack this classical view. If you look at the fundamental laws of, say, chemistry or thermodynamics, they rely completely on statistics. How does order come out of that?
Well, order only emerges from the collective, disorderly behavior of absolutely vast numbers of microscopic particles.
So you can't predict any single one?
No. You cannot predict the motion of any single atom in a gas or a fluid. It's random. It's constantly jiggling because of thermal energy. But when you assemble an enormous quantity of these atoms, the law of large numbers just smooths out all that individual randomness.
And you get these utterly predictable, lawful, macroscopic outcomes.
Exactly. Order emerges from the sheer averaging of chaos.
So the chaos is essential, but it has to be on a huge scale, where the individual fluctuations just become, meaningless noise.
Precisely. Schrödinger used this wonderful analogy of fog sinking.
Right.
An individual water droplet is undergoing this unpredictable, erratic Brownian motion. It's just jittering randomly.
It's from the thermal agitation.
But the fog mass as a whole sinks in this orderly, predictable fashion. Why? Because the movements of billions upon billions of droplets average out statistically. Chemical diffusion works the same way. Random molecular movement, yet the net result is always a uniform, predictable distribution.
That's classical physics.
That's classical physics.
But life, biological systems, they seem to operate outside of this statistical safety net. And that's the core of the challenge he posed.
It is the great tension of the book. Schrödinger observed that the incredibly precise order that defines life, the complexity and durability of an organism, is determined by genes, by what we now call chromosomes.
Okay, but the problem, as he saw it back in the 1940s, was that genes are tiny.
Very tiny. We're talking about structures comprising maybe only 1 million atoms or even less. And this size, he claimed, is much too small for the law of large numbers to apply.
So if a gene is that small, it should just be instantly overwhelmed by molecular turbulence.
Yes. The internal thermal noise of the cell, which is operating at body temperature, should constantly jiggle and disrupt the structure.
So according to classical statistical laws, it should mutate wildly, degrade. It would make heredity impossible.
It should. Yet, as he pointed out, genes are remarkably stable and persistent, a stability that he described as bordering on the miraculous.
He needed a really good analogy to drive home just how durable this molecular blueprint was.
And that's where the famous anecdote comes in, the Habsburg lip.
It's such a potent example.
It really is. He uses this distinctive protruding jaw, a complex, molecularly determined trait that was faithfully preserved in the Habsburg ruling family for centuries.
Surviving all this continuous, noisy molecular turbulence inside the body.
It's a trait that connects the grand sweep of royal European history with the stability of a tiny molecule.
So the puzzle is really simple. How can such a tiny collection of atoms reliably maintain such complex, durable order over vast stretches of time, basically violating the statistical expectations of physics?
And Schrödinger's conclusion was that life must be, in his words, guided by a mechanism entirely different from the probability mechanism of physics.
His solution was this revolutionary switch.
From order from disorder to the order from order principle.
And this is where his physics background gave him the conceptual breakthrough, or at least the theoretical framework he needed for the gene.
He hypothesized that the gene must be a rigid, large molecule, stabilized not by statistics, but by strong quantum-derived forces.
He specifically referenced the Heitler-London theory of the chemical bond from 1926.
Yes, which explains the stabilizing forces that lend rigidity to solids. In short, the gene is stable because it behaves like a crystal.
But not a boring crystal, that's the key. That's where the term aperiodic crystal comes from.
Correct. A normal crystal, like salt or quartz, is repetitive. It's a dull, predictable sequence of repeating units, like wallpaper or a repeating musical pattern.
And if the gene were that simple, it couldn't possibly encode the vast, intricate information needed for an organism.
So therefore, the gene must be aperiodic, non-repetitive. It has to be complex but structured, like a magnificent, intricate tapestry or a detailed novel.
It shows elaborate, coherent, meaningful design, not just dull repetition.
And this aperiodic structure, it provides the immense combinatorial richness, the variety of unique arrangements required to contain all that detailed hereditary information.
It's the idea that a small number of base units, when you arrange them in a non-repeating sequence, can contain enormous complexity.
And this structural hypothesis led him directly to his second great concept, the hereditary code script.
That structural hypothesis was genuinely transformative. It forced biologists to look for a rigid, stable molecule that could hold vast amounts of complexity. But we should move now to interrogate the concept of the code script itself, because that's where the deep philosophical determinism of the book really takes root.
That's right. The code script is where structure translates into instruction. Schrödinger argued that the aperiodic structure was essential because it provided the variety, like using Morse code's dots and dashes, to specify the entire plan of development.
Okay, but we have to clarify a historical confusion here. His code script was not the genetic code we understand today.
Not at all.
Not the specific three-letter cipher relating nucleotides to amino acids.
No, that misunderstanding came later. Schrödinger's code script was a philosophical and conceptual precursor. It was much broader, much more ambitious. It was a rule code, a program.
A program containing complex instructions that stipulated the structure, the function, the developmental timing of the entire future organism. It was the master plan.
And that sounds inherently, almost aggressively deterministic.
It is the ultimate expression of biological determinism, and it implies three key philosophical theses that really provided the foundation for early molecular biology's worldview. The first is what you'd call neo-performationism,
which rejects the idea that an organism is built up progressively.
Right. Instead, the full organism is, in essence, pre-specified and just unfolds.
Precisely. Schrödinger stated that the chromosomes contain, in some kind of code script, the entire pattern of the individual's future development.
So the life process, from conception to death, is just an unfolding, an acting out of what's already precisely written and fixed in the genome.
Your destiny is set at the molecular level.
And the second thesis is developmental computability, which really hammers home that extreme deterministic view.
This is where he brings in the famous concept of Laplace's demon.
Right.
Laplace, back in the 19th century, he hypothesized that as some intellect knew the exact position and momentum of every particle in the universe, it could predict the entire future.
And Schrödinger applied this directly to the embryo.
Directly. Because the developmental process is contained in the fixed, stable structure of the chromosome, he argued that a sufficiently powerful mind could, in principle, compute the entire organism.
He said it could predict whether the egg would develop into a black cock or into a speckled hen, into a fly or a maize plant.
And then there's the third thesis, genetic animism, which gives the gene itself a kind of intentional, causal authority.
It's not just a blueprint, it's the builder too.
Right. It grants the gene, the code script, both law code and executive power, or architect's plan and builder's craft in one. The gene isn't just inert information. It's endowed with active causal agency. It initiates, dictates, directs, and controls development.
And this highly reductionistic, deterministic stance, he didn't just invent it.
No, he adopted it from earlier influential geneticists like H.J. Muller, who had already defined the gene as the ultimate basis of life and the primary agent dictating an organism's features.
So Schrödinger basically reinforced and amplified an already existing powerful strain of deterministic genetics. It aligned perfectly with his larger philosophical goal of finding clockwork mechanisms in life.
That's the key. He selected the most deterministic viewpoint available in genetics to prove his point in physics.
But if we look at the last 80 years of discovery, the critical historical reversal is that modern biology largely rejects this extreme order-from-order principle and its deterministic theses.
It does.
So let's start with the stability puzzle. If the gene isn't intrinsically stable, what actually holds the show together?
Well, Schrödinger was brilliant in identifying the need for stability and the role of strong covalent bonds. But he was fundamentally wrong about stability being an intrinsic property of the crystal-like DNA molecule itself.
So what is it then?
The stability we observe, the reliable replication and repair, is a continuous, high-energy accomplishment of the living cell.
It's a process, not a state.
Precisely. DNA is just as susceptible to damage and stochastic effects from thermal agitation as any other molecule. The cell has to work tirelessly, consuming immense energetic resources.
Deploying what you called an entire army of proteins.
Exactly. Proofreading enzymes, repair complexes, all just to continuously minimize molecular noise and correct errors. If the cell dies, the DNA immediately begins to decay.
So instead of the gene imposing its crystalline stability on the messy cellular context,
The cellular context imposes stability on the messy gene.
that completely flips the causal relationship on its head.
It does. And that undermines the three deterministic theses completely. Take neo-preformationism and developmental computability. They're flawed because molecular processes, including gene expression, are fundamentally probabilistic, not deterministic.
Okay, so how does that look practically? How does this stochasticity ruin Laplace's demon?
Well, it happens at the most basic levels. The initiation of transcription, whether a gene is turned on or off, is often a stochastic event. It's influenced by local molecular noise, not a precise switch flip.
It's not a clean on-off.
Not at all. And protein folding isn't always identical. Minor variations in the folding landscape, driven by thermal noise, mean that proteins from the exact same sequence might acquire slightly different conformational states, which then impacts their function. We talk about noise in gene expression.
So even if the code script itself were perfectly known,
If the output is inherently noisy, predicting the trajectory of every single cell is impossible. The developmental instructions are probabilistic tendencies, not deterministic mandates.
so the organism becomes a product of dynamic construction, influenced by chance, rather than a perfect reading of a pre-specified blueprint.
Yes, and finally, genetic animism fails because DNA is not an inherently active molecule. It just sits there inertly in the nucleus until the already organized living cellular context activates it,
Activated by transcription factors.
which are often themselves products of earlier stochastic processes. You see the problem? It takes a cell to make a cell. And beyond that, we now recognize that order is also transmitted non-genetically.
Through epigenetic inheritance.
Right, where structures like membranes, organelles, protein complexes, they all serve as templates for their own replication, independent of the DNA sequence.
So the molecular revolution successfully implemented the structure of the aperiodic crystal, but it rejected the philosophical engine, that radical determinism that was supposed to drive it.
In many ways, yes. They swapped the rigid crystal for a dynamic, error-correcting system. The order-from-order principle, while it was conceptually inspiring, was empirically just too brittle and reductionistic.
And that realization forces us into our next section, where we have to challenge Schrödinger's outright dismissal of the statistical approach.
He claimed order from disorder was irrelevant to life.
But if we broaden our view, it becomes obvious that statistics and probability are absolutely foundational to biology at larger scales.
If you just step back from the single cell to the population, the entire engine of evolution relies on order from disorder.
Charles Darwin's theory of evolution by natural selection.
It's fundamentally statistical. Population genetics, pioneered by thinkers like Fisher and Galton, and ecological modeling with equations like Lotka-Volterra, they find predictable, lawful order, an order-producing process in the collected behavior of large numbers of individually unpredictable organisms.
So you don't need deterministic individual fate to explain why a population evolves predictably.
Not at all. The law of large numbers operates beautifully across generations of organisms.
And even within the individual organism, the concept of order from disorder has been powerfully reclaimed.
Particularly in the study of development, through the idea of determinacy in the gross.
This is a critical concept from the developmental theorist Paul Weiss. So what does determinacy in the gross mean, and how does it challenge Schrödinger?
Paul Weiss was one of the foremost critics of this kind of reductionist genocentrism. He argued that while individual molecular events and single-cell fates during development are inherently noisy and stochastic,
unpredictable at the microscopic level,
the collective behavior of that cell ensemble results in predictable macroscopic patterns.
That's a really powerful idea. The overall organ always forms correctly, even if the path taken by any single cell within it is random.
Right. Think about how a hand forms. The precise movements of every cell migrating or differentiating might be governed by noisy molecular cues, but the overall structure, 5 fingers in the right configuration, is highly determined.
So macroscopic order arises despite microscopic disorder, not because of a precise genetic mandate overcoming it.
Exactly. It's the opposite of Schrödinger's assertion that life must be strictly deterministic at the molecular scale. The organism essentially absorbs and averages out the noise.
And Schrödinger's view of order from disorder was, I suppose, constrained by his adherence to classical statistical mechanics.
It was. He didn't foresee the breakthrough concepts in self-organization that showed new, non-classical ways that order can emerge from disorder.
Like Alan Turing's work, which is so often cited in biology, even though Schrödinger missed it.
Turing's 1952 paper, The Chemical Basis of Morphogenesis, introduced the reaction diffusion model. He showed that spontaneous formation patterns like spots, stripes, or spirals could arise purely from the interaction of 2 chemical molecules diffusing at different rates.
Without any pre-existing template.
Without any pre-existing template or complex genetic instructions dictating the pattern. This is order emerging from disorder in a non-statistical, entirely spontaneous way.
That directly undermines the need for a comprehensive genetic blueprint for every single morphological feature.
Right. The gene just needs to produce the initial diffusing substances, and the physics does the rest. It was a radical shift.
Suggesting that the complexity we see in nature, like the patterns on a jaguar or the stripes on a zebra, can be generated by simple, local rules.
And while, yes, complex gene cascades were later discovered, real-life Turing systems are now being identified. They show that physical processes of self-organization contribute significantly to development.
In things like the formation of skeletal structures.
Or the ridges in the mammalian mouth palate, for instance.
And this naturally moves us to the even broader thermodynamic context, the idea of dissipative structures.
This is the critical modern framework that really replaces Schrödinger's limited view of thermodynamics. Organisms are open systems,
constantly exchanging matter and energy with their environment.
And by doing that, they maintain these low-entropic, highly ordered, steady states by perpetually staying far from thermodynamic equilibrium.
And Ilya Prigogine, who formalized this concept. Prigogine demonstrated that macroscopic patterns of order can arise spontaneously in these far from equilibrium conditions through nonlinear molecular interactions.
So the cell itself, and all its complex subcellular structures, like the mitotic spindle or complex protein folds, are examples of self-organization arising from intrinsically stochastic processes that are continuously fed energy.
Its life is a constantly sustained fire, maintaining its structure through flux. This is a crucial form of order from disorder, vital for life's maintenance, which Schrödinger just did not and could not foresee with the physics available to him.
And this brings us to that famous phrase and the enduring historical confusion, debunking the thermodynamics myth surrounding Schrödinger. This persistent claim that what is life is a foundational text in biological thermodynamics. It's one of the biggest historical inaccuracies surrounding the book.
He only dedicated a few pages to it.
A few pages, mainly to show that organisms compensate for the second law of thermodynamics entropy by importing what he called negative entropy.
Or free energy. And the concept itself wasn't even new, was it?
Not at all. Scientists like Hermann Helmholtz and Lord Kelvin in the mid-19th century had already articulated this concept, that life maintained its order by consuming external energy and that equilibrium was synonymous with death.
And the term negative entropy itself was famously criticized for being technically imprecise.
Linus Pauling, another Nobel laureate, was particularly scathing. He called Schrödinger's application of the term vague and superficial.
He argued Schrödinger should have just used the technically correct term free energy.
Should have. But more crucially for our historical understanding, Schrödinger's conception was later misleadingly conflated with the modern, rigorous concept of self-organization.
So you're saying the self-organizing theorists, like Prigogine, were retroactively tied to Schrödinger to borrow his scientific authority.
Precisely. Prigogine himself was forced to explicitly differentiate his concept of order through fluctuations from Schrödinger's much older Boltzmannian view. Prigogine's models were built on non-equilibrium conditions, which Schrödinger simply didn't address.
It was a clear historical maneuver by later scholars.
To position Schrödinger as a patriarch of non-equilibrium thermodynamics, lending enormous authority to a field that was otherwise struggling for legitimacy.
That distinction is vital. It shows that while the thermodynamic claim is really a historical myth, the true lasting influence of Schrödinger's deterministic premise on molecular biology, the mechanical view of life, is completely undeniable.
And that's what we need to focus on next. The core conceptual shift wasn't from thermodynamics to genetics. It was from a biochemical, sort of colloidal view of the cell to a rigid, deterministic, machine-like view.
We moved from order to the idea of organization from information.
The moment molecular biology adopted information talk, code, program, control it, solidified this mechanical framework.
And that terminology was heavily influenced by the rise of cybernetics.
Right, pioneered by Norbert Wiener. Wiener explicitly linked information to the negative of entropy, or negentropy, a concept he credited to Schrödinger's discussion of negative entropy. So the linguistic and conceptual seeds were sown.
And even though Shannon's rigorous information theory failed to translate smoothly into biology, it focuses just on syntax, not semantic meaning.
The metaphorical power of the code was just too strong to resist.
And the semantic shift was completed in 1953.
When Watson and Crick famously suggested that the precise sequence of the bases is the code which carries the genetical information, and then Crick solidified the deterministic flow with a central dogma in 1958. Information flows unidirectionally from DNA to RNA to protein.
Which perfectly mirrored Schrödinger's code script. The fixed sequence holds the instruction, the law code, and executive power.
And so molecular biology's foundations were essentially Schrödingerian extensions, built on two key postulates, the first being informational specificity.
This is the assumption that the meaning, the functional instruction, resides purely in the linear sequence of bases or amino acid residues.
Schrödinger relied on this combinatorial richness to justify his aperiodic crystal, and Crick formalized it with the sequence hypothesis.
And the second postulate, which connects right back to the aperiodic crystal's design, is structural specificity.
This is the assumption that for reliable, specific lock and key interactions, which are crucial for proteins to function as enzymes, machines, complex builders, the macromolecules must be rigid and stable.
Like a solid or a crystal.
Exactly. This structural rigidity protects the system from stochastic noise and thermal agitation, allowing the cell to be characterized. As Nobel laureate Bruce Alberts described it, as a collection of protein machines and solid-state circuits.
It's a marvelous engineering metaphor. It replaced that older, messier biochemical view that saw the cell more like a dynamic, statistically governed colloidal suspension with something much cleaner, predictable.
And, crucially, computable. This machine metaphor became utterly dominant. It was canonized in seminal texts, perhaps most famously in Jacques Monod's Chance and Necessity.
Monod was a biologist by training.
But he shifted entirely to this mechanical view, crediting Schrödinger for emphasizing the necessity of stability and rigidity. He noted that the ribosome appears to work mechanically, like a clock or a precision machine tool, rather than statistically.
And this is where the philosophical license comes in. The assumption of determinism and rigidity gave molecular biologists the justification they needed to just ignore the reality of molecular fluctuations and noise.
It offered a simplifying lens. If the cell is a perfect machine, you don't need to worry about noise. You just focus on the wiring diagram and the components. And the success of this model was massive.
It led to sequencing, structural biology, countless medical breakthroughs.
And that raises a crucial critical question. If the deterministic model led to the central dogma, and the central dogma allowed us to develop gene therapies and understand fundamental processes, wasn't Schrödinger empirically correct in choosing that simplifying perspective, even if the underlying physics was technically flawed?
That is the central paradox. It was an incredibly fruitful, simplifying metaphor.
The problem arises when that metaphor becomes a conceptual prison. We are now in the 21st century, facing what you could call the looming crisis of mechanistic biology.
Where experimental evidence is mounting that the foundational assumptions of rigidity and specificity are often inadequate for explaining how life actually works.
What's the evidence specifically challenging this clockwork machine view?
It starts with the molecules themselves. Experiments show that many proteins are not rigid, stable solids. They are often soft, fluid, and stochastically alternate between multiple states. They're not fixed machine parts.
And a huge percentage of the proteome, sometimes up to 40% in humans, consists of intrinsically disordered proteins, or IDPs.
These proteins lack a fixed, stable 3D structure, yet they're vital for signaling and regulation.
A machine made of soft, floppy parts that constantly change shape, that completely violates the structural specificity postulate.
Completely. Furthermore, the lock-and-key interaction model, which requires hyperspecificity, is often replaced by interactions that are promiscuous, opportunistic, and based on probability.
We're seeing evidence that large, complex assemblies, such as the ribosome itself, don't execute precise mechanical movements. They function via biased random walks, where noise is actually incorporated into the function.
So it's not a perfect machine.
No, it's a system designed to tolerate, and sometimes even utilize, internal noise.
So the deterministic machine metaphor, which allowed for enormous success, is now proving inadequate to explain the dynamic, fluid reality of the cell.
It provided the philosophical license to simplify the problem, but now we have enough data to see that the simplification missed a key element of life: the robust management of inherent molecular stochasticity. The legacy is one of both brilliant conceptualization and profound ongoing conceptual cost.
And this leads us to the heart of the matter, the philosophical stake that was really being debated back in 1944, uncovering Schrödinger's true motive for writing this book.
This is the crucial context that makes What is Life so fascinating. Schrödinger's engagement with biology wasn't purely about genetics. It was directly tied to the fierce interwar physics debates over the interpretation of quantum mechanics.
He was deeply disillusioned, even hostile, toward the orthodox Copenhagen interpretation.
The one championed by Niels Bohr and Werner Heisenberg, which mandated philosophical indeterminacy and the collapse of the wave function upon measurement.
The very interpretation that his famous dead and alive Schrödinger's cat thought experiment was designed to mock as a reductio ad absurdum.
Exactly. He was a champion of classical determinism and the idea that physics ultimately should describe reality precisely. He became particularly alarmed when other prominent physicists started extending quantum indeterminacy into biology and psychology.
Who is making this connection between quantum physics and free will?
The most direct intervention came from Pascal Jordan, a key proponent of quantum field theory. Jordan developed what was called the amplifier theory, which claimed that the random, causal behavior of individual atoms,
quantum indeterminate,
was somehow magnified within the delicate structures of organisms, providing a physical foundation for free will. Bohr also argued for a kind of complementarity. You could describe life deterministically via physics or teleologically via psychology, but never both simultaneously.
So Schrödinger saw biology becoming a proving ground for the very indeterminacy he was battling in the world of physics.
That was his true guiding motive. What is life was designed as a powerful philosophical intervention. His central aim was to demonstrate, using the genetic structure as evidence, that life is a strictly deterministic phenomenon.
Protected by quantum stability from molecular noise.
Right. This stable aperiodic crystal was intended to sever the proposed biological link between quantum mechanics and the philosophical problem of free will.
He used genetics as a highly effective weapon in a physics war. The stakes were nothing less than the philosophical direction of all of science.
And we have archival evidence confirming this strategic intent. In his correspondence with the geneticist C.D. Darlington, Schrödinger harshly criticized his physicist colleagues for being illegitimate and mischievous in trying to extract free will from quantum indeterminacy.
He stated clearly that, "I believe that with regard to living matter, the outstanding feature we are called upon to understand and to explain is the incredibly strict determinacy."
For him, quantum theory was necessary, but only to protect the hereditary substance against disorder, not to amplify it.
And this explains the famous, often baffling epilogue on determinism and free will, which wasn't even part of his original public lectures.
The epilogue was the absolutely necessary philosophical cleanup operation. Having established the scientific case for biological determinism, he couldn't just ignore the persistent human experience of agency and free will.
So he forced the question outside the domain of science altogether.
Retreating into the metaphysics of Hindu Vedanta.
Why Vedanta specifically?
The philosophy of Vedanta provides a solution by dissolving the self. It posits that individual consciousness is an illusion, and that we are all aspects of a single, eternal, universal consciousness.
So the feeling of free will, the I that makes a choice, is not a scientific or material reality.
It's a delusion of the single consciousness experiencing the world through billions of individual bodies. By proving biological determinism, he pushed the question into mysticism, effectively neutering the physicist's claims. Muller, quite famously, mocked this conclusion as straight old-fashioned mysticism.
But the strategy worked. The scientific community, especially molecular biologists, adopted the deterministic clockwork view. Jordan's speculative quantum biology faded into irrelevance.
Schrödinger's theoretical assumption provided the essential philosophical underpinning for molecular biologists to ignore quantum effects and focus purely on the mechanistic, deterministic aspects of the gene for the next 80 years.
And that brings us to the synthesis of this entire journey. We started with a book widely misunderstood as merely a collection of prescient biological guesses about DNA structure.
And we found that it was, in fact, a meticulously constructed philosophical argument aimed at preserving a classical, deterministic worldview in the face of quantum uncertainty.
We traced how his order from order principle became the foundation for molecular biology's informational specificity, giving rise to the pervasive molecular machine metaphor, a metaphor that yielded tremendous scientific success.
But we also rigorously identify the profound weaknesses in that deterministic view: the neglect of stochasticity, the reality of dynamic self-organization through principles Schrödinger didn't anticipate, and the critical dynamic role of the whole cell in actively maintaining stability.
So critiquing Schrödinger isn't an act of historical pedantry, it's an exercise in reflecting on the current conceptual limitations of molecular biology.
The book remains profoundly relevant today because its philosophical foundations, rigidity, and determinism are now actively being challenged by mounting evidence of molecular fluidity, noise, and nonlinear emergent order.
The gene is not an inert, rigid, aperiodic crystal. It's a dynamic resource, constantly battling and negotiating the fundamental forces of disorder maintained by the cellular system itself.
Schrödinger's genocentric deterministic view was a profound, powerful choice, fueled by his desire to defeat philosophical indeterminacy. And he largely succeeded in banishing that concept from biology for 80 years.
But if we look at the evidence today where we confirm the fundamental stochasticity and non-rigidity of cellular life, and where the engineering metaphors built on Schrödinger's premises are increasingly failing to capture that dynamic reality,
The question remains, did Schrödinger win the argument against indeterminacy prematurely? If, as some predict, the field of quantum biology is reborn, seriously exploring how complex, delicate quantum phenomena like coherence or entanglement might play a non-trivial, non-deterministic role in life. For example, in processes like odor reception or enzyme function. Must we, 8 decades later, finally concede that Bohr and Jordan, however speculative they were, might have been looking in the correct, unpredictable direction? It suggests that the boundary between the deterministic machine and the unpredictable living cell might be far more complex and indeterminate than Schrödinger ever intended.